Hey guys, tonight we begin somewhere deep inside you, beneath your bones, beneath your breath, where trillions of cells are quietly humming with instructions. You can't feel it, but right now your body is reading a script, not a metaphorical one, a literal chemical script, twisted into a double helix, curled up like a lazy ribbon on a gift you never asked for. Your DNA is reciting the same lines it's rehearsed since the moment you were conceived. In this story, DNA isn't just a molecule, it's a memory vault, a coiled-up code that knows how tall you'll grow, whether cilantro tastes like soap, and how your skin reacts to sunlight. It holds the chapters of your past, scribbles potential endings, and lets your body improvise the rest. And here's the kicker, you didn't write a word of it. So before you get comfortable, take a moment to like the video and subscribe, but only if you genuinely enjoy what I do here. Also, drop your location and local time in the comments. It's always fascinating to see who's drifting off with us from across the world. Now dim the lights, maybe turn on a fan for that soft background hum, and let's ease into tonight's journey together. You're gliding through the microscopic space between skin and bone, where cells glisten like underwater lanterns. Inside each one is a nucleus, and in that nucleus, the blueprint of you. You float closer, watching as the chromatin, those unraveled threads of DNA, breathe and twitch like seaweed. If you could stretch out all the DNA in your body, it would span twice the diameter of the solar system. And yet here it is, curled delicately inside a cell so tiny, thousands could fit on a pinhead. It's poetic, really. The entire map of your body, its strengths, flaws, secrets, packed into something smaller than a speck of dust. You watch the strands tighten into familiar double helices, spiraling endlessly. This isn't just structure, this is storage. A molecular hard drive. DNA is what lets your body build itself, maintain itself, and sometimes destroy itself in painfully specific ways. Scientists often call it the code of life, but let's be honest, it's more like a recipe book written by a chef who doesn't follow measurements and occasionally adds chili just to see what happens. Still, every living thing you've ever known, plants, whales, that weird fungus in your shower, runs on some version of this code. It's the closest thing nature has to a universal language. Just 4 bases, A, T, C, and G, but when arranged in just the right way, they can make a banana or you. Now here's something wild. Your DNA is mostly inert. Most of it isn't doing anything right now. It's coiled and resting, like a script waiting for the right scene. A small portion, though, is actively being read by tiny molecular machines, ribosomes, enzymes, transcription factors, that glide along the DNA like factory workers on an assembly line. They're making proteins, building blocks, signals, defenders, messengers. Everything that keeps you alive is part of this non-stop production cycle that starts with DNA. It's oddly comforting, isn't it? You might feel like a chaotic bundle of thoughts and emotions up top, but deep down your body is calm, rhythmic, consistent. DNA never panics. It doesn't get insecure or distracted. It just does. And it remembers. Every cell, well, almost every cell in your body carries the exact same DNA you started with. Muscle, skin, neurons, blood, they all hold identical copies, but they read different parts of the script. Your eye cells read the chapter on pigment. Your pancreas reads the chapter on insulin. It's like giving 200 types of actors the same script and telling them to perform completely different plays. This brings us to a lesser-known tidbit. Some cells go rogue. Yes, your body contains cells that ignore the program. Cancerous cells are infamous for this. They mutate, misread the script, refuse to die when they should. It's a genetic mutiny. Scientists still argue whether it's purely a coding failure, or if environment, the set, the stage, the lighting, pushes them off course. But before we spiral into doom, let's rewind. Picture this. Your DNA isn't just about you. It's a mosaic made from your parents. Half from your mother, half from your father, but not in a perfect 50-50 split. The way your chromosomes shuffled during conception is like a genetic poker game, random, high stakes, and completely out of your control. Sometimes you get lucky, sometimes you inherit the family curse. There's something strangely humbling about it. Your sense of identity, your laugh, your mood swings, your crooked pinky finger, isn't solely yours. It's borrowed, inherited. A remix of ancient scripts that go back thousands, even millions of years. And just to spice things up, your DNA includes viruses, Yep, about 8% of your genome is made up of viral DNA, leftovers from ancient infections. These retroviruses inserted themselves into our ancestors' reproductive cells, and now they're part of the furniture. Most are harmless now, but they're there, like forgotten tenants in your genetic apartment building. Imagine that. You're part human, part virus. Your cells are like libraries, with ancient dusty scrolls that say "do not read" in faded ink. But sometimes, the body reads them anyway. As you hover near a glowing strand of DNA, you notice something else. It's damaged, a slight twist, a broken rung on the ladder. Radiation, toxins, even time itself causes wear and tear. Luckily, your cells have repair crews, enzymes that patch, glue, and splice with eerie precision. But they're not perfect. Some errors slip through. And just like a typo in a novel, some change nothing, while others rewrite the ending entirely. So here you are, a walking, talking novel, a first edition print with missing pages, smudged ink, and a few glorious, unrepeatable typos, and it's still being written. Every time a cell divides, it copies your DNA. Every copy is slightly different, like making a photocopy of a photocopy. Eventually the image degrades. This is one reason you age. There's even a structure called a telomere, tiny protective caps on the ends of your chromosomes that shorten over time. They're your body's internal hourglass. When they run out, the cell stops dividing. Game over. Still, DNA is durable. It's outlived mammoths, frozen in permafrost. It's been found in teeth, bones, hair, hundreds of thousands of years after death. That means one day, long after you're gone, someone might read your script, not for your story, but to understand theirs. You hover now at the molecular level, staring at the biological equivalent of alphabet soup. A, T, C, G. Just 4 letters, 4 chemical bases. And yet somehow, this pitifully small cast of characters writes the entire script of life on Earth. Kind of insulting, right? You'd expect at least a full alphabet, maybe some punctuation. But no, just 4. Adenine, thymine, cytosine, guanine. They pair up across the rungs of your DNA ladder. A always with T, C always with G, like the most loyal dance partners in the universe. Millions, even billions of these pairs. Stacked, twisted, repeated, rearranged. Now here's the weird part. Those letters don't do anything themselves. They're just instructions. What actually gets built from them, what your body actually uses, are proteins, enzymes, hormones, structural stuff. Everything from melanin in your skin to the dopamine scrambling across your brain to make you feel vaguely interested in this video. And the order of those letters? That's everything. You rearrange just a few, and suddenly, instead of digesting dairy, you don't. Or your hair grows curly instead of straight. Or you're allergic to cats. Or if your genetic recipe is particularly spicy, you can eat entire habanero peppers without blinking. This isn't hyperbole. Your tolerance for spicy food might actually be partially genetic. Scientists are still digging into the specifics, but sensory sensitivity is encoded right there in your DNA. So next time someone mocks your spice tolerance, just whisper, "It's in my genome." The joke is, it probably is. But beyond party tricks, these 4 letters shape everything. They control the shape of your red blood cells, whether you'll get male pattern baldness, what your voice sounds like, even your tendency to wake up at 6 a.m. like a cursed Victorian ghost. And that's just the obvious stuff. There are genes for how efficiently you metabolize caffeine, for how impulsive you are with money. There's even emerging research connecting specific genetic variants to how easily you feel lonely. Imagine telling your ancestors that somewhere inside you, a strand of molecules can make you more likely to overthink a text message. Still, it's important to say, genes aren't fate. They're blueprints, not blueprints with contracts attached. Your DNA might hand you a loaded gun, but whether it ever goes off depends on environment, chance, and a dozen invisible factors you can't control. Scientists still argue how much power our genes really have over our behavior. Some say it's just raw potential, like seeds that may or may not grow depending on the soil. Others argue that your genes quietly steer everything, like a subtle but stubborn current you can't quite swim against. But everyone agrees on this. You're not just your DNA, you're how it's read, when, where, and how often. Take something as simple as eye color. You might think it's a brown or blue binary. It's not. It's a web of over a dozen different genes, interacting like a jazz band with no conductor. And depending on which parts are activated, you could end up with stormy gray, sea glass green, or hazel that changes with the light. This is where the alphabet soup gets tricky. You see, only about 1–2% of your genome actually codes for proteins. The rest used to be called junk DNA. Scientists thought it was just filler, a biological accident, like scribbled out lines in an old manuscript. They were wrong. We now know this so-called junk contains regulatory sequences, enhancers, silencers, basically backstage crew. They tell the protein coding parts when to perform. It's like discovering the useless backstage pass is actually the key to the whole show. Even more bizarre, some of these non-coding sequences seem to function like ancient commentary tracks, remnants of genes that once worked in long-extinct ancestors. Others are involved in how tightly DNA wraps around itself, which directly affects how genes are expressed. In short, most of your DNA doesn't code, it coordinates. Think of your genome as a massive symphony. Only a few instruments play the melody. The rest are rhythm, harmony, timing, echo. But if any of them play the wrong note, the whole thing turns into a mess. And yes, every person's song is different. Even among humans, your genome is about 99.9% identical to everyone else's. That 0.1% difference is where everything personal lives. Your height, your laugh, your weird mole, your love of pineapple on pizza. That's it. That 0.1% makes you you. Now zoom out with me. Way out. Beyond your skin, your city, your planet. Every living thing on Earth shares DNA. You share a third of your genes with daffodils, about 60% with fruit flies, 98.8% with chimpanzees. You and your goldfish? Over 70%. And yet we think we're special. Maybe we are, but the ingredients list says otherwise. And still, despite this elegant simplicity, DNA can't explain everything. Identical twins have the same genome, but they age differently. One gets a disease, the other doesn't. One becomes a CEO, the other grows bonsai trees in rural Kyoto. Why? Because even with the same letters, the story can play out differently. Context changes meaning, and that's where we're headed next. But before we go, one more strange fact to tuck under your pillow. There are people, very rare people, who have more than one genome. It's called chimerism, usually the result of absorbing a twin in the womb. That's right, some people are walking around with two completely separate sets of DNA in their bodies, one in their blood, another in their skin, or even half their organs coded by a different script. So if you ever feel like there's another version of you living inside, there might be. You drift now toward the very beginning, before your name, before your first breath, before your mother even knew you were there. It starts with 2 cells. Just 2. One from your mother, one from your father. A sperm and an egg, each carrying exactly half the genetic blueprint needed to build you. Each packed with potential, like 2 USB drives colliding in the dark. And then, contact. One furious, desperate little sperm breaks through the egg's surface. The DNA inside both cells unspools. 2 half-scripts fuse. And just like that, a new genetic story begins. Yours. This is the zygote. You at your most microscopic. Just a single cell, quietly pulsing in the warm dark. And yet inside that one cell is the entire instruction manual for your body. Height, skin, allergies, mental health vulnerabilities, hairline fate. All of it. No pressure. It's strange to think about now, but this first cell already contains a complete genetic identity, an irreversible code. There's no second draft, no rewrites. It's locked in. From this point on, you are a first edition with one shot. The zygote doesn't waste time. It divides. 1 cell becomes 2, 2 become 4, 8, 16. Soon it's a snowball of cells tumbling through the fallopian tube, each one faithfully copying your DNA. Well, mostly faithfully. Every time a cell replicates, it copies about 3 billion base pairs. That's like retyping the entire King James Bible with mittens on. Sometimes, the cellular copy machine makes a typo, a misspelled gene here, a duplicated sequence there. Most of the time, your body catches and corrects these, but occasionally the mistake stays, and that's how mutations begin. Not all mutations are bad, though. Some do nothing. Some make you lactose intolerant or super taster sensitive to bitter foods. Some, in rare cases, might even offer a strange evolutionary edge. Scientists still debate whether early embryonic mutations, those first few typos, could quietly shape a person's entire future. They've found that even identical twins can differ in early cell divisions, setting them on subtly different biological paths before they're even born. You float closer to the early embryo now, a hollow ball of cells called a blastocyst. It's oddly elegant, like a tiny soap bubble. This is when things start getting interesting. The cells begin to specialize. Some decide they'll become brain. Others choose to be heart, skin, spine. And all of this, still guided by DNA. Imagine a crowd of people holding identical scripts. But some are highlighting chapter 4, others are skipping to chapter 12. That's what's happening in your body. All your cells share the same genetic code, but they express different parts of it depending on their job. This process, cell differentiation, is part miracle, part chaos. The embryo doesn't have a master architect. It's more like a jazz improvisation. Genes turn on, others off. Some take cues from nearby cells, others seem to act on hunches, and in the middle of it all, you, or the rough draft of you at least. Now let's zoom in on one of those cells. You'll notice something strange. There's a gene getting turned off, not deleted, just silenced. This is called genomic imprinting, and it's one of the earliest forms of epigenetic editing in your life. See, not all genes are treated equally. For some, only the copy from your mother or your father gets used. Never both. The other one just sits there. Scientists still argue over why this happens. Some say it's evolutionary conflict. Your mom's genes want to conserve resources. Your dad's want you to grow big and strong. Others think it's random noise in the genetic system. Either way, it means some parts of your genetic story are censored before you're even born. And here's something quirky. Some of those early choices are permanent. If a gene is silenced in your early embryo, it may never get activated in your entire life. It's like tearing a page out of a book before it's even published. As the embryo grows, layers form, germ layers, 3 of them to be precise: endoderm, mesoderm, ectoderm. From these humble sheets, everything emerges: lungs, heart, bones, nervous system. They twist and fold into shape like origami guided by invisible hands. And still, every twist is dictated by that original DNA script. But remember, DNA doesn't operate alone. It's reading cues from the environment, the chemical soup surrounding the embryo. Hormones, temperature, even signals from the mother's immune system influence which genes get turned on. This is where the idea of nature versus nurture gets messy. Nurture starts in the womb. Long before you experience love or language or schoolyard trauma, your developing cells are already negotiating with their environment. Some of these negotiations leave marks. There's a study where pregnant women who experienced famine during the Dutch hunger winter gave birth to children whose genes had been chemically altered in utero, changes that made their bodies store fat more efficiently. Those children, decades later, had higher rates of obesity, heart disease, and diabetes. That's how early your DNA starts writing, and how early life starts editing. So even if you've never known hunger, your DNA might remember. A ghost of famine passed down through molecules. Now let's dip into a lesser-known fact. During development, there's a brief window where your body builds both male and female reproductive structures. Only later, based on hormonal and genetic cues, do some dissolve and others grow. But remnants can linger, tiny leftover tissues that never activate. Your body, in a way, starts with options. It chooses one path, silences the other, and continues forward. But sometimes the decision isn't so clean. There are rare conditions where the body reads mixed signals. Chromosomes say one thing, hormones say another. The result? Intersex variations. Not a mistake, just another version of the story. A reminder that biology doesn't always care about binaries. By the time you're a fetus with fingers and a heartbeat, your genetic story has already laid down the major plot points. But the details, how tall, how smart, how sensitive, how at risk for depression or Alzheimer's or celiac disease, those are still being sculpted. You don't get a second draft, but you do get edits, side notes, footnotes, margin scribbles, and by the time you're born, Your DNA has already written the first chapter. You're older now, still floating through your body, but no longer inside the soft blur of embryonic development. You've grown, your cells have matured, and now something remarkable is happening behind the scenes. Not all of your genes are active. Wait, let's back up. Remember how every cell in your body has the same DNA, like a shared script given to thousands of actors? Well, the real magic lies in how your cells decide which parts of that script to perform. This is gene expression, and it's not just a binary thing, on or off. It's a dimmer switch, a volume knob, a timing cue. Genes aren't read in full like pages of a book. They're sampled, skipped, trimmed, or amplified, depending on the cell's mood and mission. You walk into the kitchen. Your stomach cells are making enzymes to digest dinner. Meanwhile, in your skull, neurons are busy manufacturing dopamine, so you can feel a vague sense of reward from finishing that Netflix episode. Same DNA, different readings. Now, what decides which genes are flipped on or off? That's where transcription factors come in. Tiny molecular bouncers that let some genes enter the club and keep others waiting in line. These molecules bind to specific regions on your DNA, often upstream of the gene itself, acting as switches. Some of them are always active. Others are triggered by hormones, nutrients, temperature, or even stress. Yes, stress. You stub your toe, cortisol floods your system. That hormone travels into the nucleus of your cells and flips specific genes into gear. Suddenly, your body's inflammation response is activated. White blood cells get mobilized. Your blood sugar rises, all because a switch was flipped. It's unsettling how responsive your genome is, how little it takes to rewrite your internal chemistry. And here's where it gets even weirder. Your cells remember. This isn't science fiction, it's cellular memory. Let's say a skin cell is exposed to UV radiation. It activates a protective gene response. If that happens enough times, that skin cell's descendants will remember the exposure and stay on alert. It's like passing down a family story, only instead of a campfire tale, it's instructions for how to survive future sunburns. And these switches, they don't just flip once and stay that way. Many genes turn on and off multiple times a day. Your circadian rhythm, for instance, your internal clock is largely driven by gene expression cycles that loop every 24 hours. So if you've ever felt like a different person at 2 a.m. versus 2 p.m., that's because you are. Gene expression is in flux all the time. What you eat, how you sleep, whether you exercise, how lonely you feel, it all talks to your genome, constantly. This is where the metaphor breaks down. DNA isn't a static script, it's a living score, a jazz piece being played and replayed, reinterpreted moment by moment. Scientists still argue how permanent these expression changes can be. Some seem to last only hours. Others, especially during early development or trauma, can stick for decades. In extreme cases, they can even be passed down to your children. You drift through your liver now. It's a genetic powerhouse. Liver cells are masters of adaptation. They adjust their gene expression based on everything from alcohol to medication to viral threats. You ate fast food 3 nights in a row, your liver flipped on detox genes. Took a new medication, different genes. That celebratory whiskey shot, more gene switches. Your DNA doesn't change, but how it's used shifts constantly. Now let's throw in a fun oddity. Some cells in your body express genes they don't need, just to help other cells. Take lactating breast cells. They crank out huge amounts of gene products, proteins, antibodies, not for themselves but for a baby. It's biological altruism. Your DNA stepping up, not for your survival but someone else's. Then there are genes that stay off forever, like the ones your body used to build you in the womb. Once the scaffolding is done, those genes shut down, forever. They're like construction crews that pack up and disappear, leaving only traces behind. But sometimes genes that should be silent wake up. This is how some cancers start. A gene meant to be dormant in adult life suddenly reactivates. It starts instructing cells to divide, divide, divide. No breaks, no pause, just endless duplication. And here's where it gets terrifying. The same gene in another context might be completely harmless. It's not the gene that's dangerous, it's the switch. You pass by a neuron now. These cells rarely divide, but they're busy in other ways. Their gene expression is tailored for memory, emotion, thought. One of the most active genes in your brain is called BDNF, brain-derived neurotrophic factor. It helps grow and connect neural pathways. The more you learn, the more BDNF gets expressed. It's literally writing brain structure based on your experience. And guess what boosts BDNF? Exercise, meditation, deep sleep. So yes, when people say running clears their mind or sleep helps them focus, that's not woo-woo. That's gene expression at work. A fun side note, there's a gene variant called ACTN3, sometimes dubbed the sprinter gene. People with this variant are more likely to have explosive muscle fibers, making them better at power sports. If you've ever wondered why you suck at long-distance running but love sprints, that might be why. Still, genes are no guarantee. Plenty of people have the right variants and never use them. Others don't, and outperform expectations anyway. DNA hands you tools. Whether you pick them up, that's on you. And that brings us to one of the most unsettling and beautiful truths about gene expression. You're shaping your body in real time. Every laugh, every workout, every heartbreak leaves a molecular trace, not just in your memory, but in how your cells read your code. You are not just written by your DNA, you're also the editor. You drift now into the space between the genes, the vast, quiet stretches of your genome that don't code for anything, at least not in the way you think. Welcome to the so-called junk DNA. For decades, scientists dismissed this genetic filler as leftover debris, relics from evolution with no real purpose. After all, only about 1% of your DNA actually codes for proteins. The rest? They figured it was like static between radio stations. Dead air, noise. Turns out that noise is doing a lot of whispering. As you glide along the helix, you start noticing something peculiar. In these long, non-coding stretches, there are strange regulatory elements, enhancers, silencers, insulators, little molecular road signs. They don't build proteins, but they control the parts that do, like a director who never steps on stage but dictates every cue. These sequences tell your cells when to activate certain genes. Where, how much, for how long. They're like behind-the-scenes stagehands keeping the spotlight exactly where it needs to be. And sometimes these quiet areas of DNA mutate, and when they do, it can wreak havoc. There are diseases, like certain forms of cancer or congenital heart conditions, that are linked not to errors in a gene itself, but to glitches in the switches that control it. A gene might be perfectly fine, but if its enhancer misfires, chaos unfolds anyway. Here's the kicker. A single gene can have dozens of these control elements. They might be located far away, thousands of base pairs upstream or downstream, like long-distance relationships trying to manage a performance together. And sometimes, they fight. Yes, parts of your DNA are in conflict. Some sequences push a gene to express, others push it to stay quiet. The final expression isn't a binary yes or no, it's a vote. A noisy molecular democracy where context is everything. Now let's venture into an even stranger part of your genome, transposons, also known as jumping genes. These are sequences of DNA that can move around, literally. They cut themselves out of one place and paste themselves into another. Some are ancient viral hitchhikers, others are endogenous, part of your genome's natural chaos. Roughly 45% of your genome is made up of these mobile elements, nearly half. And they don't just sit there quietly. Some still jump. Some insert themselves into active genes and disrupt them. Others trigger immune responses, or influence how nearby genes behave. Scientists still argue whether these elements are mostly harmful, neutral, or secretly useful. Some theories suggest that jumping genes might be a hidden engine of evolution, introducing the randomness needed for innovation. Others think they're freeloaders, existing simply because your body hasn't figured out how to get rid of them. But here's the twist. Your body does try to silence them. There's a process called DNA methylation, a chemical tag that attaches to these sequences and says, essentially, shoo, stay quiet. Methylation is one of the main ways your cells keep rogue sequences from going off script. It's also one of the key mechanisms behind epigenetics, which we'll get into in the next section. For now, understand this. What we once thought of as genetic garbage is more like dark matter, unseen, mysterious, powerful. Your non-coding DNA might not make proteins, but it makes decisions. Even your chromosomes, those iconic X-shaped bundles, are held together and folded in part by non-coding sequences. The way your genome physically folds inside the nucleus changes how it's read. Two genes might be thousands of letters apart, But if the DNA loops a certain way, they can end up side by side, affecting each other. It's like origami with consequences. This spatial organization is why your DNA isn't just a linear string of code, it's a 3D object, a twisted, folded sculpture that controls access to its own information. And yes, when this folding goes wrong, diseases can emerge. There's even a term for it: chromatin remodeling disorders. Your genome's architecture matters as much as its content. Now let's throw in one more curveball: long, non-coding RNAs. These are RNA molecules transcribed from your so-called junk DNA. They don't become proteins. Instead, they float around the cell, interacting with DNA, RNA, and proteins in subtle ways, sometimes silencing genes, sometimes activating them, sometimes regulating entire networks. In 2012, scientists discovered a specific long non-coding RNA called X-IST, which is responsible for silencing one of the two X chromosomes in females. Without it, the dosage of X-linked genes would spiral out of control. So yes, your junk DNA helped figure out dosage compensation before your high school math teacher ever could. You float through these non-coding regions now, watching molecular machines move silently through the loops and folds. It's a ghost town, a whisper network, a hidden stage behind the curtain of the genome. And somewhere deep in this space, there are pseudogenes, broken copies of real genes. They look like functional genes, but have been deactivated by mutations over time. Scientists used to think they were meaningless leftovers, but new research suggests some pseudogenes still do things. They might regulate their functional cousins. They might soak up molecules that would otherwise silence other genes. They might be decoys, or backups, or something else entirely. Again, scientists still argue, but the takeaway is clear. Your genome is more than a list of instructions. It's an ecosystem, a balance of noise and function, of code and commentary, of actions and options. What we once called junk might be the reason you're alive right now. So next time you think about your DNA, remember, the story isn't just in the words. It's in the margins, the silences, the skipped lines. You're made not just from what's written, but from everything that isn't. You're floating now just above your own genetic manuscript, but something's changed. The letters are still there, A, T, C, G, repeating endlessly, but there are scribbles in the margins, tiny chemical flags, stickers, smudges, notes in the margins that weren't there when the story began. This is epigenetics, the science of how your environment, behavior, and even emotions can modify how your DNA is read without changing the actual code. Think of it like sticky notes on a script. Skip this line. Read this part louder. Ignore this character completely. The original words remain untouched, but the performance changes. And those notes, they're written by life. Every stressful deadline, every warm memory, every sleepless night, every skipped meal, it all leaves its mark. Your DNA doesn't exist in a vacuum. It responds. It adapts. It takes notes. The most common tools for this scribbling are methyl groups and histone modifications. Methylation is like a tiny chemical lock. When a methyl group attaches to a gene's promoter region, it can silence that gene. No code gets read. No protein gets made. Histone modifications, on the other hand, adjust how tightly DNA is wound. Imagine your genetic material as a spool of thread. The tighter it's wound, the harder it is to access the information. Loosen it, and the cell can read it more easily. You're not just a passive product of your genes. You're constantly rewriting how those genes express themselves. Now here's where it gets existential. Your grandmother's trauma. It might still be echoing in your body. Studies in mice have shown that exposing a parent to a traumatic experience, say the smell of a certain chemical paired with a shock, can cause their offspring to react fearfully to that same smell, even though they were never exposed to it. Scientists still argue whether this happens in humans too, but the early evidence is chilling. It suggests that your DNA remembers not just biology, but experience, that your cells are carrying more than just instructions. They're carrying stories. Some of them aren't even yours. Let's ground this with a modern example. Stress. When you're under chronic stress, your body floods with cortisol. That cortisol travels through your bloodstream, enters your cells, and influences which genes get expressed. Some of those genes affect inflammation, metabolism, mood regulation. Long-term stress can literally reprogram how your body handles threat. Even worse, if you're stressed during pregnancy, the fetus gets that biochemical message, and it may adjust its own gene expression accordingly, gearing up for a dangerous world before it even arrives. This is called fetal programming. And it means that your genes are constantly eavesdropping on your environment and adjusting your settings accordingly. Now, not all epigenetic changes are bad. Some are protective. Exercise, for example, can switch on genes involved in muscle repair, memory, even immune strength. Mindfulness practices have been linked to gene expression patterns that reduce inflammation. Certain foods like cruciferous vegetables or green tea contain compounds that promote healthy methylation patterns. So yes, your choices matter. You're not doomed by your DNA, you're in dialogue with it. But let's get weird again, because you knew this was coming. Some epigenetic changes are permanent. They don't just affect you, they can be passed on. Your grandfather smoked heavily. Some research suggests his grandchildren might have a higher risk for asthma. Your mother endured famine or war. Her epigenetic responses might shape your stress hormones. This is called transgenerational epigenetic inheritance. And while scientists still argue over the full extent of it in humans, animal studies make it hard to ignore. In other words, you may be living out reactions to events you never personally experienced. Your DNA isn't just your own, it's haunted. Now for a quirky side note, bees. In a beehive, all the female bees have the same DNA, but some become sterile worker bees, and one becomes the queen. The difference? Royal jelly, the food fed to developing queen larvae, silences certain genes and activates others. Same genes, different expression. One becomes royalty. The others? Not so lucky. Your body works the same way. Two people can have the same risk gene for, say, type 2 diabetes, but only one develops it because of diet, exercise, stress, or even gut bacteria. Epigenetics is the reason one person thrives and another struggles, despite having near-identical blueprints. But there's a darker edge to this too. There are epigenetic changes you can't reverse. Exposure to lead, benzene, or certain endocrine disruptors, especially in early life, can silence or activate genes in ways that persist for decades. Some researchers believe this may help explain the rise of certain cancers, autoimmune conditions, or even mental illnesses in specific populations. And here's where things get complicated. We don't have a full map. We're still learning what these changes mean, which are harmful, which are neutral, which are trying to help us. It's a field in progress, a scribbled notebook that humanity is just starting to decode. But what we do know is this: your DNA is not a static command center. It's a responsive, evolving participant in your life. It listens, it remembers, it adjusts. You are not just the sum of your genes. You are the sum of how your genes are read, rewritten, and sometimes silenced. You are a manuscript constantly in revision. And right now, this moment, this breath, your choices are influencing the script. So maybe drink some water, maybe take a walk tomorrow. Maybe remember that stress isn't just mental, it's molecular. And maybe, just maybe, forgive yourself for the pages you didn't write. You've landed now in a place that defies logic. You're floating between two people. Identical twins, same DNA, same face, same laugh, but under the hood, entirely different stories. They started from the same zygote, a single fertilized egg that split into two embryos. Genetically, they are clones, yet as they grow, subtle differences emerge. One develops a peanut allergy, the other doesn't. One becomes anxious in crowds, the other thrives on stage. So how does this happen? How can two people with the same genetic script perform such wildly different plays? Here's the short answer: gene expression. But you already knew that. The longer answer is more intimate. Even before birth, these two developing humans encounter slightly different microenvironments. One might get more nutrients through the placenta. One might experience more stress from a momentary shift in blood flow. These tiny differences kick off a cascade of epigenetic changes. Methyl tags get added or removed. Histones shift. Genes flick on or off. And over time, those flicks become habits. Habits become identities. You float now between their neurons, tracing how one twin's brain lights up at the smell of cinnamon, while the other remains indifferent. It's subtle, but it's real. Now let's fast forward. One twin moves to a polluted city. The other stays in the countryside. 10 years go by. Their lungs, livers, and immune systems are reading the same DNA, but expressing different sets of genes to cope with their environments. The city twin develops asthma. The country twin doesn't. Same code, different life. And the longer they live, the more those differences accumulate. This phenomenon is so well documented that researchers now study epigenetic drift, the gradual divergence of gene expression in identical twins over time. By the time they hit 60, the similarity in their epigenetic markers can drop dramatically. Sometimes they're no more alike than siblings. But it's not just the environment. It's also random chance. Cells make mistakes. They flip the wrong switch, or they misplace a methyl group. And those little errors over decades create divergence, like two copies of the same book where the pages yellow at different speeds, stain in different places, and fall apart in different hands. And here's something odd. Even identical twins can have different numbers of chromosomes. It's rare, but during development, cells can divide unevenly. One twin might have a mosaic condition, a mix of normal and altered cells, while the other doesn't. You wouldn't even know by looking at them. But deep inside, their genetic story has forked. And yet here comes the twist. Even with all this divergence, identical twins often report feeling a deep, unspoken connection, finishing each other's sentences, feeling each other's pain, sometimes dreaming the same dreams. Scientists still argue whether this is biology, psychology, or something stranger, but it reminds us that genes don't just shape the physical. They influence perception, personality, and intuition. Now imagine you had a twin. How would you feel knowing someone out there shares your exact genetic code but not your life? Would it feel like a mirror or a ghost? Some identical twins say it's like seeing the version of yourself that got away. Others say it's like sharing a consciousness with someone who took a different exit ramp. And the more we study twins, the more we realize just how flexible our genome is. It's not the DNA alone that makes you who you are. It's how it dances with the world. Let's talk about another kind of twin, conjoined twins. These are identical twins who never fully separated during development. They can share limbs, organs, even parts of the brain. And yet, despite their shared body, they often have entirely separate personalities, preferences, dreams, desires. It's a haunting reminder that identity doesn't live in the genome. It lives in expression and in experience. So here's a lighter tidbit. Even when twins are genetically identical, their fingerprints are different. Why? Because fingerprints are shaped not just by DNA, but by pressure, movement, and blood flow in the womb. Tiny shifts in the uterine environment leave a permanent mark. Literally. You are shaped by randomness as much as by code. Now back to your body. No twin required. Inside you, every organ is reading the same genome in its own way. Your brain and your stomach are working off the same playbook, but interpreting different chapters. That's why some people develop brain-based disorders like schizophrenia, while their genetically identical twin does not. Or why one twin might get breast cancer and the other doesn't. The blueprint is never the full story. Your genome is a deck of cards. You get dealt a hand. But the game you play depends on the table, the lighting, the noise in the room, and whether someone spills their drink on your cards halfway through. And sometimes, the dealer makes a mistake. There are cases of vanishing twins. Early in pregnancy, two embryos start developing. Then one is absorbed into the other. The surviving baby is born seemingly alone. Until years later, doctors find traces of a second genome in their body. A patch of skin, a testicle, a tooth in the brain. Yes, that happened. It's called chimerism. And it's not just rare, it's reality bending. There are documented cases where someone's blood DNA doesn't match the DNA in their saliva, or where a woman gave birth to a child who didn't match her genetics until they tested her cervical tissue and found her vanished twin's DNA. You think you're one person, but genetically, you might not be. And even if you are, you're still 1,000 versions of yourself across time. Baby you, teen you, heartbroken you, rebuilding you. They've all left marks on your body, on your cells, on your genome. You don't need a twin to see how stories can diverge from the same script. All you need is time. You're floating now along the fragile, glittering edge of your own DNA strand. But something looks off. One of the base pairs, normally locked in its partner's embrace, has slipped. A rung in the ladder is cracked. A letter has changed. Welcome to the world of mutation. A mutation is, at its core, a typo, a small error in copying. A C becomes a T. A segment is deleted. A chunk is duplicated. Sometimes it's like autocorrect gone wrong, turning hello into hell. Other times it's a harmless swap color to color, changing nothing meaningful. And yet, sometimes that typo rewrites the entire ending. Mutations happen constantly. Your cells divide trillions of times over your life, and each time they must replicate around 3 billion letters of DNA. Even with proofreading enzymes and backup systems, the margin for error is inevitable. On average, Each newborn baby has about 100 to 200 brand new mutations that weren't present in either parent. Most of these do absolutely nothing. They land in non-coding regions, or they're redundant thanks to the genetic code's built-in wiggle room. But some are game changers. You drift into a red blood cell gene, specifically the one that produces hemoglobin. A single letter mutation here can cause sickle cell disease. Just one switch from A to T alters the shape of the protein. The red cells become sickle-shaped, sticky and brittle. Now here's the wild part. In certain parts of the world, that same mutation offers protection against malaria. The misshapen cells don't host the parasite as easily. So is the mutation a flaw or an adaptation? Scientists still debate this, but the answer is probably both. Nature doesn't care about perfection. It cares about survival. Mutations are a gamble, sometimes devastating, sometimes brilliant, often both. Let's zoom out. You float toward the gene responsible for the protein P53, the guardian of the genome. This protein prevents damaged cells from dividing. If this gene mutates, that safety check disappears. Cells begin to replicate uncontrollably. In other words, cancer. And yet, in elephants, yes, elephants, there are 20 copies of the P53 gene. That's one reason why, despite their enormous size and billions of cells, they have a surprisingly low rate of cancer. A genetic redundancy built by nature to prevent disaster. Now here's something quirky. Some people have mutations that give them superpowers. Not comic book powers, mind you, but subtle advantages. There are rare individuals who don't feel pain, others who sleep deeply on just 4 hours a night thanks to a mutation in the DAC2 gene. There are people with unusually dense bones, withstanding impact better than most, or those with hyper-efficient muscles, faster healing, stronger hearts. They exist, walking anomalies, hidden among us. Of course, the flip side is far more common. Mutations linked to cystic fibrosis, Huntington's disease, BRCA breast cancer, Tay-Sachs, whole sections of the genetic script that misfire catastrophically. Some mutations are inherited, passed quietly from parent to child like a loaded dice. Others happen spontaneously, during replication, exposure to radiation, even during the moment of conception. And it's not just humans. Mutations drive evolution across all life. They're the raw material for change. Giraffe necks, bird beaks, antibiotic-resistant bacteria, all sculpted by the randomness of error, then shaped by selection. Mutation is how life experiments. Now float into the mitochondria, those little power plants inside your cells. They have their own DNA, separate from the nuclear genome. Mitochondrial DNA is inherited only from your mother, and it mutates faster than the DNA in your nucleus. This means researchers can use mitochondrial mutations like time-stamped breadcrumbs to trace human migration over tens of thousands of years. It's how we discovered mitochondrial Eve, a woman who lived around 150,000 to 200,000 years ago, and whose mitochondrial lineage survives in all living humans today. And here's the kicker. Her DNA didn't survive because she was the only woman. It survived because her descendants kept having daughters. That's it, a genetic roll of the dice that played out for millennia. Mutations also explain why no two people are genetically identical, unless they're identical twins. Your genome is 99.9% the same as the person next to you, but that 0.1%, that's roughly 3 million differences, most of which came from mutations passed down through generations. You are, in a very real sense, an accumulation of mistakes that worked. But not all mutations are ancient. Some happen in your lifetime. Somatic mutations, those that occur in individual cells, can lead to cancer, gray hair, new moles, or shifts in immunity. Scientists still argue how often these mutations affect behavior or personality. Some suspect certain neurological conditions like autism or schizophrenia may involve subtle somatic mutations in the developing brain, but proving it is hard. The brain doesn't exactly offer up tissue samples willingly. And then there's the ultimate mutation experiment, radiation. High doses can shred DNA, but even low doses, like cosmic radiation faced by astronauts, create mutations. That's one reason long-term space travel remains so risky. The farther you get from Earth's magnetic shield, the more your DNA is exposed. If humanity ever settles on Mars, our genome will change, slowly, unpredictably, and forever. And speaking of the future, there's a mutation spreading right now in parts of the human population. It's called the CCR5 del32 deletion. People who inherit 2 copies of this mutation are resistant to HIV. Their cells don't produce the receptor the virus needs to enter. It's not science fiction. It's happening. Evolution, in real time. And it raises a big question. Should we encourage these mutations? Use gene editing to spread the good ones? Erase the bad? That's where we're headed next. But before we go, remember this. You are the result of billions of years of trial and error. Every cell in your body is a compromise, a risk, a bet placed long before you were born. And somehow, here you are, still mutating, still writing, still alive. You're floating now in the blood, the warm red river that carries oxygen, hormones, and memories of people long gone. Not metaphorical memories, actual fragments, echoes etched into your DNA. Because your genome isn't just a record of you, it's a historical document, a time capsule stitched together from ancestors you'll never meet, many of whom didn't even consider themselves fully human by today's standards. Let's talk about the ghosts in your blood. Roughly 1–4% of your DNA is Neanderthal. If you're of non-African descent, that is. It's not just an interesting trivia fact. It's an inheritance. Those genes may influence your immune system, skin sensitivity to sunlight, pain threshold, even how your body processes fat. You carry the remnants of a long-extinct species in your cells, not just as fossils in a cave, but as code, still active, still working, and it doesn't stop there. If you have ancestry from Southeast Asia or Oceania, you might also have traces of Denisovan DNA, another mysterious hominin species that interbred with early humans. These genes have been linked to high-altitude adaptations found in modern Tibetans. In other words, you can breathe thin air because someone thousands of generations ago had a forbidden forest hookup with a Denisovan. Romantic, right? This is introgression, when genetic material from one species gets absorbed into another through interbreeding. And it's not rare, it's rampant. It means that your genome is a mosaic, not a single line of ancestry, but a spiderweb of merging stories. You didn't just inherit your grandmother's nose, you inherited entire evolutionary experiments. Some genes that help you fight viral infections today, they came from Neanderthals. A few that may increase your risk for depression or autoimmune conditions, also Neanderthal. You float now into one of your chromosomes, looking for a sequence that never belonged to your species. It's still here, quiet, functional, like an old lamp wired into a modern house. Nobody questions it, but if you trace it back far enough, you realize that part of your genome was built by someone else. It's humbling. You are not a purebred anything. You are a remix. Even the Y chromosome passed from father to son carries evidence of deep splits and remerging over tens of thousands of years. It's how geneticists traced back male ancestry to a single common ancestor, nicknamed Y chromosomal Adam. No, he wasn't the first man, just the lucky guy whose Y chromosome didn't go extinct. Meanwhile, mitochondrial DNA passed from mother to child, tells its own story, separate from the rest of your genome. By studying its mutations over time, scientists have built a map of ancient human migration, revealing how Homo sapiens expanded out of Africa and slowly, stubbornly populated the planet. You are the endpoint of that journey. Your bones, your skin, your brain, all engineered by generations of survival, adaptation, chance, and love. Every scar, every instinct, every allergy is part of that story. And here's something else. You don't just inherit genes, you inherit gene variants, subtle tweaks that evolved to help your ancestors survive in their unique environments. If your ancestors lived near the equator, your skin developed melanin to shield you from solar radiation. If they lived in colder, cloudier places, they likely evolved lighter skin to better synthesize vitamin D from weak sunlight. There's a reason some people can digest lactose and others can't. It tracks back to ancient herding cultures that began drinking animal milk. In those populations, a mutation emerged to keep the lactose enzyme active into adulthood. Every trait you have was earned or failed to be removed by thousands of years of pressure and probability. Scientists still debate how many of these ancient adaptations remain relevant. Some clearly do, like high-altitude respiration or malaria resistance. Others, not so much. Your body may still respond to stress like it's being chased by a lion, even if it's just an email notification. That mismatch between ancient genes and modern life is what some call evolutionary lag, and it's probably why chronic stress, obesity, and autoimmune disorders are so prevalent now. Your DNA is optimized for a world that no longer exists. Let's get quirky again. Did you know red hair might have Neanderthal origins? Some studies suggest certain variants of the MC1R gene came from our extinct cousins. That means every time someone makes a ginger joke, they're probably insulting an ancient hybrid. Nice. And then there are genes that defy geography. In 2020, scientists found a gene variant common in modern Tibetans that helps them survive at high altitudes. But it didn't evolve in Homo sapiens. It came from Denisovans. You're breathing thanks to someone your ancestors once considered other. So much for tribalism. You are at the molecular level, a monument to mingling, and you're still mixing. Genetic variation continues today, across borders, languages, cultures. Every child born is a fresh combination, a new shuffle of the ancestral deck. And here's the strange comfort in all of this. You're not separate from history. You are history. Your DNA carries the footprints of migrations, wars, famines, love stories, genocides, survival. Everything humans have ever endured or celebrated is inside you, in fragments, in echoes, in silent code. And one day when you're gone, your DNA might still be here, in a child, a database, a fossil, a hair in a time capsule drifting through the solar system. You are not the beginning. You are not the end. You are the middle of something very old that keeps writing itself forward. You're no longer floating through your cells now. You're hovering above a crowded bar, a high school dance, a dating app interface, a candlelit dinner. Wherever attraction happens, DNA is quietly at work. Because let's be honest, sex is biology with better lighting. But it's not just about pleasure. It's about the survival of your genes. You might think you're swiping left or right based on charisma, looks, or a decent Spotify playlist. But deep down, something else is scanning. Immune system compatibility, genetic diversity, fertility potential. You are a vehicle for your DNA, and your DNA wants to make more of itself. This brings us to sexual selection, the evolutionary process, where traits are favored not because they help you survive, but because they help you reproduce. Peacocks have ridiculous tails, not for camouflage, but because peahens like them. That's it. Tail dates. Humans are no different, except our displays are more subtle. Confidence, humor, bone structure, pheromones, bank statements. Some researchers believe you can smell genetic compatibility. It's called major histocompatibility complex, MHC detection. The MHC is a set of genes involved in immune function. People tend to be more attracted to the scent of someone with different MHC genes because genetic diversity means better immune defense and potential offspring. You're not just picking someone who feels right. You're picking someone whose immune system balances out yours. That cologne you like might just be an unconscious signal from your MHC. And then there's facial symmetry. It's often called a marker of genetic health, even though scientists still argue about how important it actually is. But symmetrical faces tend to be rated as more attractive across cultures. Your brain is wired to see balance and assume stability. Even height and voice pitch play a role. Deep voices in men, associated with higher testosterone levels. Certain waist-to-hip ratios in women, correlated with fertility. We dress it up in poetry and romance, but much of attraction is just ancient code running in the background. And don't forget behavior. Dominance, kindness, emotional intelligence. These traits signal reproductive success in different ways. Evolution has shaped not just what you look for in a partner, but how you behave to attract them. The flirt, the flex, the eye contact, all of it has genetic fingerprints. Now let's zoom in on sex itself. The moment a sperm meets egg, your DNA gets a lottery ticket. That fusion creates a unique genetic combination. Half from one, half from the other. The chromosomes mix, recombine, shuffle like a deck of cards dropped in midair. And sometimes, the shuffle includes mutations. Tiny errors that might become irrelevant, or turn into traits, or spark an illness, or even rarer, offer a survival advantage. This is evolution's gamble. Roll the dice every generation and see who thrives. Some of these mutations become widespread, others vanish. The process is slow, brutal, and relentless. It's not about perfection, it's about persistence. You think you chose your partner, but your DNA was choosing too. Let's talk survival. Once offspring exist, natural selection takes over. Traits that help survival, strong immunity, cleverness, adaptability, get passed on. Traits that don't, well, nature doesn't send a sympathy card. But here's where it gets psychological. Some of the traits we consider useless or even harmful in modern society may have once served a reproductive purpose. Take risk-taking. In ancestral environments, it might have helped with hunting or defending a tribe. Today it might look like speeding or starting a tech startup. Either way, it signals something primal. Confidence, potential, boldness, Same goes for traits like jealousy. Evolutionarily, it may have protected against mate poaching, ensuring you pass on your genes. Today, it causes fights at brunch, but the wiring is ancient. There's even a theory called costly signaling, the idea that showing off traits that require energy or risk proves you have enough resources to waste. Think expensive watches, flamboyant dancing, or elaborate mating rituals on The Bachelor. Yes, you're watching a biology contest in cocktail dresses. Now here's a strange twist. Not all sexually selected traits are helpful long-term. Some may increase reproductive success while reducing health or longevity. Think testosterone-driven aggression. It might attract mates, but it also increases risk of injury or early death. Reproductive success and survival don't always align. Evolution doesn't care if you live long, just long enough. And sometimes traits persist because of frequency-dependent selection. A trait might be rare and attractive now, but if everyone starts having it, its value drops. Think blue eyes, or a unique sense of humor, or being the only one who doesn't use dating apps. So where does this leave us? Floating through a world where DNA is constantly matchmaking. You think you're choosing someone who fits your vibe, but deep down your genome is scanning for diversity, health, and maybe a bit of novelty. Not because it loves variety, but because genetic recombination thrives on difference. Even sexual orientation has genetic threads. It's not a binary switch, but a complex weave of genes, hormones, and prenatal development. Scientists have found correlations with certain genetic markers, but no gay gene. The current consensus? It's biological but not easily reducible, and reproduction isn't even the whole story. Not everyone wants children. Not everyone can have them. And yet DNA still plays a role in desire, drive, attachment, and bonding. Traits that once served evolutionary goals now shape how we love, stay, or leave. Which brings us back to the uncomfortable truth. Your DNA doesn't care about your happiness. It cares about replication. That's why you're wired for desire, but not necessarily for contentment. Why falling in love feels like a drug, but long-term happiness takes work. One ensures you mate. The other? Optional, as far as evolution's concerned. So next time your heart races around someone new, remember, your DNA is whispering, yes, them, that one might make good copies, but don't take it too seriously. You're more than a gene delivery system. You get to choose how your story plays out, even if your genome has opinions. You're drifting now into unfamiliar territory, soft, humid, microbial. You've left your own genome behind and entered something far stranger, the other genome inside you, your microbiome. At this very moment, your body is home to trillions of microorganisms, bacteria, viruses, fungi, all coexisting, mostly peacefully. They outnumber your human cells. Seriously, there are more of them than you, and they bring their own genes. In fact, the combined genetic content of your microbiome is hundreds of times larger than your human genome. Some scientists even refer to this collective as your second genome. It doesn't just coexist with your biology, it co-writes it. Let that sink in. Your body is not a single organism, it's an ecosystem. You hover now inside the gut, the grand central station of microbial life. Each person has a unique composition of gut flora, influenced by diet, birth method, environment, and even pets. Your microbes help you digest food, absorb nutrients, and synthesize vitamins. But their role goes far deeper. Gut bacteria produce neurotransmitters like serotonin, dopamine, and GABA. Over 90% of your body's serotonin is made in your gut, not your brain. The phrase gut feeling, yeah, that's not just poetic, it's biochemical. Your microbes can influence your mood, memory, even decision-making. Mice raised without a microbiome behave differently, less anxious, more risk-taking. When given microbes from anxious mice, they become anxious. The gut literally talks to the brain via the vagus nerve. Scientists still argue about the full extent of this gut-brain axis. Some call it a revolution in neuroscience. Others caution that correlation isn't causation, but the evidence is mounting. Your microbes whisper to your neurons, you are not alone in your own mind, and the effect isn't just psychological. The composition of your microbiome can affect obesity, diabetes risk, autoimmune diseases, and even how you respond to medication. Certain gut bacteria make you better at extracting calories from food. Others protect against inflammation. Some can activate or suppress genes in your immune system. In other words, your microbial tenants are rearranging your gene expression patterns from the inside, and it starts early. If you were born vaginally, You got your first dose of microbes from your mother's birth canal. If you were born via C-section, your starter pack came from skin and air, totally different microbial makeup. Breastfeeding introduces another layer. Early antibiotics? They reset the whole ecosystem. And yes, these early microbial influences affect your gene expression and immune development for life. Now here's something bizarre. There are certain gut bacteria that crave sugar. They've evolved to survive on it. And when you starve them, they produce molecules that influence your cravings, urging you to eat more sugar so they can thrive. You thought that 11 p.m. cookie craving was all you? Think again. Some scientists believe this microbial manipulation is part of an evolutionary arms race. They influence your behavior to meet their needs while you try to resist them. It's not always malicious, just deeply symbiotic. And yet this relationship can go sour. An imbalanced microbiome called dysbiosis can lead to chronic inflammation, mood disorders, metabolic issues, even colorectal cancer. Certain gut bacteria can metabolize compounds into carcinogens. Others can produce anti-inflammatory agents that suppress tumor growth. Your gut flora are not just passengers. They're editors of your health. And yes, there's still debate. The field is new, and many studies are still correlative. But every year we uncover more ways these microbes talk to your genome. They produce short-chain fatty acids that influence gene expression in immune cells. They help regulate blood sugar by modulating insulin sensitivity genes. They even influence the expression of epigenetic markers in your colon. And they're not just in your gut. You have microbes on your skin, in your mouth, inside your nose, even in your lungs. Each niche hosts its own ecosystem, and each of those ecosystems plays a subtle but significant role in how your body functions, heals, ages. Ever heard of the hygiene hypothesis? It suggests that modern cleanliness, while preventing infections, also deprives us of crucial microbial exposure early in life. This lack of training may cause immune systems to overreact, leading to allergies and autoimmune diseases. Your DNA expects to be in constant dialogue with microbes. When that conversation goes silent, your immune system panics. It attacks pollen, or peanuts, or your own tissues. Now let's talk fecal transplants. Yes, they're exactly what they sound like. Transferring stool from a healthy donor into the gut of someone with a damaged microbiome. Sounds gross. Works miracles. In cases of Clostridium difficile infection, an aggressive gut bug, fecal transplants can restore microbial balance and save lives. And with it, the genes. A new set of microbial DNA starts rewriting your gut's gene expression. It's like updating your operating system via poop. Welcome to modern medicine. And this is just the beginning. We've already mapped the human genome. The next frontier is the human metagenome, the full collection of microbial genes influencing our biology. Soon we may personalize diets and treatments based not on your genome, but on your microbiome. We may alter mood disorders by changing gut flora. We might treat obesity or depression by editing microbial genes instead of human ones. You float now back toward your own body. It's familiar again, but different. You realize you're not one story, you're two. The DNA you inherited, and the DNA you adopted. You are a walking genetic collaboration. You don't just carry genes, you host them. You're drifting now through darker currents, the kind of genetic territory people prefer not to think about. Here, DNA doesn't just build you, it betrays you. Genes can be helpful, but some lie dormant like landmines, buried deep, waiting for the right moment or the wrong one. A mutation, a stressor, a birthday. Then suddenly, they flip, and the story turns. You're passing through one of them now. BRCA1. It's a gene that helps repair DNA, an internal mechanic. But when it mutates, its protective function fails. Damaged cells multiply unchecked. The risk of breast and ovarian cancer skyrockets. Some people call it a time bomb gene. Others call it a warning. It's not alone. There are thousands of genetic variants associated with disease. Huntington's, Tay-Sachs, cystic fibrosis, Alzheimer's, hemophilia, schizophrenia. The list goes on, and not all of it is rare. We all carry about 5-10 potentially harmful mutations. Most just never get triggered. So what decides which ones do? That's where things get complicated. Take schizophrenia. There's no single schizophrenia gene. It's influenced by hundreds of variants, each one nudging the risk higher or lower. Add in prenatal development, childhood trauma, social environment, drug use, and suddenly genes become less like a sentence, more like a dice roll. Scientists still argue whether complex mental disorders are mostly genetic, mostly environmental, or some unpredictable fusion of both. Twin studies suggest a strong hereditary component, but not certainty, not destiny, which might be scarier, honestly. The idea that a subtle mix of experiences can awaken something genetic you never even knew you carried. Let's talk depression. There's no gene that guarantees it, but certain variants in serotonin transport genes like 5-HT-TLPR can influence how sensitive your brain is to stress. People with specific forms of that gene are statistically more likely to develop depression if exposed to major life trauma. Without trauma, they're often fine. Your DNA isn't a villain. It's just reactive, flexible, frighteningly attuned to your surroundings. Now float into the APOE gene, specifically the APOE4 variant. It's linked to increased risk of Alzheimer's, not certainty, risk. Some people carry 2 copies and live into their 90s with sharp minds. Others develop symptoms decades earlier. And here's something strange. In some historical environments, APOE4 may have been beneficial, enhancing cognitive function under nutrient-poor conditions. What harms us now may have once helped us thrive. DNA's not good or bad. It's context-dependent. But that doesn't stop it from hurting people. In families with known genetic conditions, choices become heavier. Do you get tested? Do you tell your children? Do you take preventive action like surgeries or medications before anything's gone wrong? These questions aren't hypothetical. They're decisions real people make every day. They're psychological burdens etched into the genome itself, and they go beyond physical illness. There are genes that affect behavior, impulsivity, aggression, addiction. Some researchers have identified variants in the MAOA gene, dubbed the warrior gene, that are linked to higher aggression in individuals exposed to early abuse. But again, it's not destiny. Abuse without the gene doesn't always lead to violence, and the gene without the abuse, often nothing. You're not a slave to your DNA, but it definitely picks the locks. Then there are the people born with chromosomal anomalies, trisomy 21, Down syndrome, Turner syndrome, Klinefelter. These are not broken humans. They're alternate expressions of the genome, different outcomes, not lesser ones. Still, society has a habit of viewing genetic difference as defect, which brings us to an uncomfortable truth. Some of history's darkest movements were rooted in a twisted view of genetics. Eugenics. The idea that we can purify the gene pool by selectively breeding or sterilizing certain populations. A pseudo-scientific horror show that led to forced sterilizations, genocide, and discrimination. We must remember genes explain variation. They do not assign value. But even today, the ethics around genetic knowledge are murky. Should insurance companies have access to your genome? Employers? Law enforcement? Did you know investigators have solved cold cases by tracing DNA through genealogy sites? They never tested the suspect, just his cousins. You are not the only person your genome affects. Now let's flip the script. There are people born with protective genes, variants that shield them from disease, like PCSK9 mutations that lower cholesterol dramatically, or CCR5 delta 32, which offers resistance to HIV. Some companies are trying to harness these protective variants, turning them into gene therapies. The idea? If you can't fix your genes, maybe you can borrow someone else's. But what happens when we start labeling people as genetically superior? We're already edging toward it. Some fertility clinics offer polygenic embryo scoring, selecting embryos based on predicted IQ, height, or disease risk. It's not science fiction. It's in brochures. Scientists still argue over how reliable these scores are, and more importantly, how ethical they are. Where is the line between prevention and preference? Between healing and engineering? You float now past one final gene, a variant in the FAH gene, associated with pain and sensitivity. People with this mutation can feel pressure, but not pain. They don't get anxious. They don't panic. They heal faster. It sounds ideal. But many of them die young, from infections, untreated injuries, or simply failing to notice when something's wrong. Pain, as it turns out, is essential. So are sadness, fear, and anxiety. Genes that make you vulnerable may also make you human. Maybe the goal isn't to edit them out, but to understand them better. You are not your weaknesses. You are how you live with them. Your genome is not a verdict. It's a narrative, and no story, no matter how heavy, is finished until the final page. You're drifting now into a different kind of terrain, not molecular, not ancestral, but modern, personal. Lit by glowing screens and curiosity, you're standing in your bathroom, spitting into a little tube, sealing it shut and mailing it to a lab. Congratulations, You've just ordered your genome. Welcome to the age of genetic testing. In the last decade, personal genomics has gone from sci-fi novelty to casual stocking stuffer. Companies like 23andMe, Ancestry DNA, and others have turned your double helix into a consumer product. For a few $100 and a little saliva, you can access a version of your code. You open the app a few weeks later. There it is, your ancestry report, a color-coded pie chart. Maybe you're 43% Scandinavian or 12% West African, or you find out you have a great-great-grandparent from an island you've never heard of, and you feel something. Pride, surprise, identity, confusion. Because DNA doesn't care about borders or culture. It doesn't come with flags or recipes, but we wrap our identity around these numbers anyway, sometimes joyfully, sometimes dangerously. You scroll further, health reports, carrier status, disease risk. Your risk for Alzheimer's is 17% higher than average. Your likelihood of type 2 diabetes is lower. You might be a fast caffeine metabolizer. You might have the sprinter gene. And suddenly the question hits, what do I do with this? Because knowing isn't the same as preparing, and risk isn't destiny. You see, most of these tests don't read your whole genome. They scan for specific variants, SNPs, or single nucleotide polymorphisms. They're statistical snapshots, not full diagnostics. If your DNA is a novel, these tests are reading it like a half-skimmed Yelp review. Scientists still argue how actionable this information really is. Take the APOE4 gene, linked to Alzheimer's. If you have 1 copy, your risk goes up. 2 copies? Much higher. But not everyone with APOE4 develops Alzheimer's, and plenty who don't carry it still do. It's a risk factor, not a prophecy. So what do you do with this information? That's where the ethical debate gets messy. Some people feel empowered. They adjust their lifestyle, eat differently, start monitoring their health more closely. Others spiral, losing sleep, fixating, catastrophizing. Information can feel like control or like a curse. And then there's family. Because your DNA isn't just yours. When you find out you're a carrier for a genetic condition, your siblings, your parents, your kids are affected too. That knowledge creates ripples across dinner tables, hospital visits, entire family narratives. Some people discover they're adopted. Others uncover long-lost siblings, biological parents, hidden affairs. The truth, when it lives in your genome, doesn't always stay hidden. In fact, the rise of consumer genetics has rewritten thousands of family stories. A woman thinks she's 100% Italian. Her results say she's half Ashkenazi Jewish. A man discovers he's not biologically related to the father who raised him. Cousins reconnect. Secrets unravel. DNA doesn't lie, but it doesn't explain everything either. Here's where it gets even more tangled. When you send your saliva to a company, you're not just buying a report, you're donating data. Many testing companies aggregate user genomes into massive databases used for research. Some partner with pharmaceutical companies. Some allow law enforcement access under certain conditions. That's not inherently bad. These databases have helped scientists identify gene-disease links, improve drug development, and even solve cold case murders. But it raises huge privacy questions. Who owns your genetic data? Can it be sold? Can it be subpoenaed? Could it be used against you? Insurance companies in some countries are restricted from using genetic data to set premiums. But not all. Laws lag behind technology. What you spit into a tube today could affect your insurability tomorrow. And even when the laws are clear, the cultural implications are murkier. Would you want to know if you had a gene linked to early onset Alzheimer's? What if there's no cure? Would you tell your partner if you found out you carried a genetic risk that could affect your children? These aren't hypotheticals. These are questions being asked in clinics and living rooms every day. And it gets stranger. Some companies now offer polygenic risk scores, calculating your genetic likelihood for things like intelligence, addiction, even income level. The science is shaky, the ethics shakier. Because what happens when we start ranking embryos by DNA? Some IVF clinics already allow parents to choose embryos with the lowest risk of certain diseases. But the temptation to take that further, to select for height, IQ, athletic ability is growing. It's not Gattaca, not yet, but we're edging closer. Scientists are divided. Some argue for tight regulation. Others say access to genetic information is empowering. Everyone agrees the tech is moving faster than our ability to ethically absorb it. You float now back to your own genome. It's tempting to think of it as a map, fixed, knowable, predictive. But really, it's more like a weather forecast: cloudy with a chance of cancer, sunny with a 14% higher likelihood of musical ability. And just like weather, prediction is not control. You might learn that your body metabolizes alcohol poorly, but that doesn't stop you from ordering another drink. You might know you carry a gene variant associated with loneliness, but that doesn't mean you'll end up alone. DNA is powerful, but it's not fate. It's a context, a set of suggestions, a user manual written in a language you're still learning to read. So if you've already tested your DNA, or if you're planning to, here's the real takeaway. Use it to inform, not to imprison. Use it to ask better questions, not to close the book, because the person you are is written in more than code. You're also made of stories, choices, memories, resilience. Your genome may write the prologue, but you still get to choose the ending. You're standing now at the threshold of something strange, something that feels like science fiction but isn't. The laboratory is cold, sterile, humming with machines, and in a petri dish under soft blue light, a strand of DNA is being edited, not nudged, not influenced, edited. This is CRISPR. If you haven't heard of it, or if you've only heard the buzzword, CRISPR is a tool, a pair of molecular scissors, a way to cut into your genetic script with shocking precision and say, let's change that word. It's based on a natural defense system found in bacteria. Scientists figured out how to hijack it to edit genes in virtually any living organism, humans included. And suddenly, a power we barely understand is in our hands. You float now over a sequence in a cell, a mutated gene, one that, left untouched, will cause a debilitating genetic disease. The CRISPR system moves in, guided by an RNA sequence matching the target, locks on, slices the DNA, and either disables the gene or replaces it with a correct version. Just like that, the script is changed. It sounds miraculous, and in many ways it is. In 2019, a young girl with sickle cell disease had her stem cells edited with CRISPR. They were re-injected into her body. She began producing healthy red blood cells. No more transfusions, no more pain crises. Her life changed. But here's the catch. That edit was in her somatic cells. It helps her, but it won't be passed to her children. There's another kind of edit, the kind that changes germline cells, sperm, eggs, embryos. These edits become permanent, heritable. They rewrite not just one life, but a family line. And in 2018, a Chinese scientist claimed to have done just that, edited the embryos of twin girls to make them resistant to HIV. The scientific community was stunned, not because it wasn't possible, but because it was done too soon, too recklessly, too unethically. Those girls weren't sick. They were test cases. We still don't know how it will affect them or their children. This is where the science gets philosophical. Just because we can edit the human genome, does that mean we should? CRISPR doesn't just correct, it enhances. In theory, we could use it to increase muscle mass, reduce anxiety, enhance memory, but what starts as therapy can become preference, and preference can become pressure. Imagine a future where parents are expected to edit embryos for higher intelligence, where the unedited are seen as irresponsible or inferior. This is not alarmism, it's a natural trajectory if left unchecked. Scientists still argue where to draw the line. Therapeutic edits, sure, but enhancement? Designer babies? It's murky. Especially when you realize that even tiny genetic changes can have unpredictable ripple effects. You edit a gene for height, but that gene also affects heart development. You tweak for memory, but now the brain is more prone to anxiety. The genome is not a list, it's a web. Tangle one strand and the rest moves. There's also the issue of equity. Gene editing is expensive. What happens when only the wealthy can afford to give their children the best version of themselves? We already have inequality built into society. CRISPR could encode it, literally, into the body, and yet there's so much good it can do. Imagine a world where no child is born with Tay-Sachs, where cancer mutations are edited out before they ever activate, where people suffering from genetic blindness or paralysis can be healed with a single cellular intervention. It's possible. But the question isn't just what we can fix, it's who gets to decide what counts as broken. Is autism a difference or a disease? Is short stature a disadvantage or just variation? Is deafness something to cure or a culture to preserve? These aren't just scientific decisions. They're social, political, personal. You're no longer just drifting through DNA. You're navigating a tightrope between progress and hubris. Here's something quirky. Researchers are now using CRISPR to engineer gene drives, genetic edits that spread through a population faster than normal inheritance would allow. The goal? Wipe out malaria-carrying mosquitoes by making them sterile. It's brilliant. But what happens when that gene drive escapes into another species? CRISPR is powerful, but power without humility is dangerous. And yet, if used carefully, it could be the greatest medical tool we've ever had. Already, scientists are working on CRISPR-based treatments for blood disorders, some cancers, HIV, and even inherited forms of blindness. You float now back into your own cells. For now, they remain untouched. Your genome is still yours, flawed, ancient, beautiful. But the future is clear. Editing is coming, fast. Soon you'll have a choice. Keep your story as it was written, or take up the pen and revise it. One day a child may ask their parents, "Did you edit me?" And the answer, whatever it is, will shape not just identity but ethics, memory, and what it means to be human. Your DNA is no longer a sacred scroll. It's an editable document. But be careful what you erase. You might delete something you didn't know you needed. You've come a long way, through replication and mutation, selection and expression, attraction, memory and error. But here you are now, standing at the end, or maybe the beginning of your own genetic thread, not just as a reader of the code, but as part of it. One more node in the massive recursive system called life. And like every system, it has to end. But DNA doesn't. When your body dies, your genes don't vanish immediately. Cells continue to transcribe for hours, some even days. Your hair and nails seem to grow, though it's mostly illusion, flesh retracting. But the DNA, it lingers. Quiet, undead. You can be long gone and still leave genetic footprints. Hair at a crime scene, bone in an unmarked grave, a saliva swab in a dusty file. Forensic labs can resurrect identity from the smallest scraps, reanimating people into evidence, family, legacy. And then there's reproduction. If you've had children, you've passed your DNA forward. Your body ends, but your code continues, spliced into a new script, edited by another parent, reinterpreted by a new generation. A remix, a sequel, sometimes a reboot. But even if you never reproduce, your DNA can live on. In biobanks, in research databases, in frozen samples, in sequencing projects that don't delete data when a person dies. Your genome could be helping cure disease 50 years from now, or being trained on by an algorithm, or floating in a capsule through interstellar space, waiting for alien archaeologists to decode it. And what about cloning? Yes, technically it's possible, not like in movies, but the tech exists. Animal cloning is routine. Human cloning is legally and ethically blocked in most of the world, but the possibility lurks. The question isn't can we, it's should we. It always has been. You might imagine someone growing a copy of you from old tissue. It would look like you, might even move like you, but it wouldn't be you, because DNA isn't the full story. DNA doesn't hold your memories, or your voice, or the way you looked out the window when it rained. DNA isn't personality, it's possibility. And yet that possibility is vast. Right now, scientists are using CRISPR to edit pig DNA so their organs can be transplanted into humans. They're resurrecting ancient genes from extinct animals. They're building synthetic genomes from scratch, writing code that's never existed in nature. You are alive at the moment in history when life itself became programmable. That should be terrifying and awe-inspiring. You carry DNA that once crawled, climbed, sailed, and wept. You carry genes from people who survived ice ages, famines, wars, and heartbreaks you'll never understand. You are a continuation of their resilience. You are not the final chapter, but you do have a say in how your line continues or doesn't. And maybe that's the point. Not legacy, not perfection, but choice. DNA gives you structure, but how you live, how you speak, forgive, love, create, endure, that's not genetic, that's you. So what do you do with the story you've been given? You could analyze it, edit it, pass it on, or just sit with it, quietly, gratefully, knowing it's a story billions of years in the making. And it chose you. So now, as your breath slows and your body begins to sink into sleep, you let go of the spirals, the sequences, the symphonies playing deep within your cells. Your DNA isn't buzzing anymore. It's quiet now, curled tightly, resting in the dark of your nucleus, like a handwritten letter folded into a drawer. Maybe you're thinking about what you carry. Maybe you're wondering what parts of you came from long-forgotten ancestors, or what echoes of you might survive long after you're gone. But for now, you don't need to solve it. You've walked the labyrinth of your genome tonight, not with microscopes or data, but with wonder. You've seen how a handful of letters can shape a universe, and how a tiny coiled thread can tell a story so old, it almost sings. And that story, it's still being written. Every choice, every experience, every quiet moment like this one, leaves a mark, not always on the DNA itself, but on what the DNA makes of you. So lie still, let the code hum softly beneath your skin. Let your body do what it's always done: repair, renew, remember, all while you dream. You are made of stardust, yes, but you are also made of stories, and your story matters, even if no one ever reads it but you.